all domains we have networks. We have business networks, social networks, chemical networks, brain networks. And this has led to the idea that there should be common approaches to different kinds of networks. And this really started in the 90s. Before the 90s, there was not enough information about networks. So we didn't know about biochemical networks, we didn't know about networks in the brain. People were analyzing social networks of Z's karate club. So it's a small karate club with 13 members, and people were spending years and years analyzing this network with 13 members. So that's all the data that was available. But in the 90s, more data was becoming available. You have all the social networks on the internet that are starting up. You have email networks. You have the internet itself as a network of individual networks that are connected. And those are some of the first articles that appeared in the 90s to analyze networks. And people found that networks are organized very differently from what they thought how they would be organized. So there were new ideas coming up in the 90s about how systems are organized. You can use those methods and apply them to many domains. One domain was the spreading of viruses, so very topical at the moment. It was applied to the spreading of SARS. You had the actual data how SARS was spreading from the origin to different parts of the world and how many subjects were infected with the virus. And this pattern could be reproduced in a computer model that was based on the airline network. So you know the flights that are going off between different airports, you know how many seats are on each airplane, you know how big the cities are that are the destination and the source of the flight. And based on this information, you can predict how many people would be infected over time. So you can reproduce the actual pattern of infection of this outbreak. You can use it for identity and search in social networks. So for example, you can look at email communication within a company. And this can lead to some surprising results. There was a big US consulting company, and they were wondering, what about our internal research office? So they basically had a research unit within the consulting company that was coming up with new methods how to talk to clients. And then they looked at the email network, and they found out that the unit is completely isolated. So it's not talking to the rest of the company. So they found that quite interesting because they spent $50 million every year on that internal unit that apparently did not communicate with the rest of the company. You can apply this to large-scale metabolic networks, so basically which molecules are interacting with which other molecules within a cell. And this can also be used to develop new drugs. There was a startup company in the UK, eTherapeutics, and they look at how the network is connected, and they try to find out what is the best way to destroy the network. So if you have MRSA, multiple resistance Staphylococcus aureus, if you have bacteria which don't respond to drugs, and you want to find out how can you knock out that bacteria, you can basically look at the network of metabolites and find out which parts should be destroyed. Another example is energy networks. There was one PhD student in the US, and that student was analyzing the electricity networks in the US. It was basically finding that there were only two lines that connect Manhattan to the rest of the electricity network. So if you cut those two lines, then there's a total blackout in Manhattan. So shortly before submitting the PhD thesis, a government agency was dropping by at the desk and saying, you get your PhD, but you're not allowed to publish the results. No one should be allowed to know what lines to cut. And network approaches have also been used for the brain. So this is the first review in the field where we looked at networks in the brain. At that point, we didn't know how the human brain is connected. We only had information about rhesus monkeys and about a small roundworm called C. elegans. But then later the term Connectome appeared. It was coined in 2005 and is now part of the Oxford English Dictionary. It's the set of connections in the brain. Connections could be between different brain regions, or they could be between individual nerve cells. So the set of connections is the connectome. So what are some of the network features of the connectome, and how do we get information about the connectome? So for example, if you think about the rhesus monkey, that's how they look like. The size is like a cat, so they're not, don't think gorilla, so they're relatively small. But how do we get information about how the brain is connected? The best way to do this is to use tract tracing. And this means that you are injecting a substance, a dye, into one part of the brain. The substance gets picked up by the cells and is then traveling from the cell body to the synapse. And we can then slice up the brain, and if we find this dye in another brain region, we know that there must have been a connection from the source to this region here. There must have been a fiber tract. Otherwise the dye wouldn't have traveled there. So this gives us very detailed information about how the brain is connected. Can we think of any disadvantage? So slicing up the brain is a problem, right? I mean, if you have a patient in the hospital and you say, well, actually, we want to know about how the brain is connected, we just need to slice up your brain. That doesn't go down well. There are some data sets in terminally ill cancer patients that agree to have this procedure just before death, but normally we just have animal data from tract tracing. Thankfully, in the mid-90s, a non-invasive way was proposed, called diffusion tensor imaging. So this is something that can be done in a normal MRI scanner. Okay, so any scanner in a hospital or research scanner, you can put someone in there for maybe half an hour, and then we can reconstruct how the brain is connected. So how does it work? What MRI is mainly measuring is hydrogen, and hydrogen mainly comes from water. So you can basically look at water in the brain, and we can look at how water is diffusing, so how water is moving over time. If there is no barrier, you start at the middle, then water could be anywhere within the sphere, okay, because it can travel in all directions. On the other hand, if you have a barrier, it might travel along one direction, but it might be blocked for another direction. So if we have a barrier for diffusion, we would get this kind of ellipsoid or cigar shape. And any idea what could be a barrier for water?
Membranes.
Membranes, yes. So in general fat. Okay, so that's why it's called white matter. So we have those connections in the middle of the brain, and they have a sheet of fat wrapped around them, a myelin sheet. Okay. And because there is so much fat wrapped around them, it appears white when you dissect the brain. So this fat is basically blocking the diffusion of water. So if you have a pattern like this one here, so in this case here, there was just water outside, so water can diffuse in all directions. You have a sphere here. And let's say you have one fiber tract in this direction, another one in this direction. Water can easily diffuse along the fiber tract, but it can't easily diffuse through those fatty membranes. So that's basically blocking the diffusion. So if you have this, you would reconstruct the network and say, well, there must be a connection in this direction, and there must be another connection in this direction here. So we can then reconstruct how the brain is connected. If you open any textbook about the brain, you will see a figure like this one here. What you see is the visual system. You have information coming from the eye, from the retina, then going over an intermediate step, a lateral genital nucleus, and then entering the cortex. So first V1, and then information is splitting up into an upper pathway, this one here. So that's for the position of objects that we see. So where in space are they and how do they move? And a lower pathway, a ventral pathway, which tells you what object is it. Is it like a cup, or is it a tire or a car? And this is the image you see in textbooks. So this is the information that undergraduates will see if they study medicine or biology or psychology. And this is the real visual system. So in reality, the network is a bit more complicated than what you see in the textbook. Can any of you immediately see the difference between the upper and the lower pathway? Yeah, just let me know, because I don't see it either. So basically, you need to use statistical tools in order to find out what pathways are there. And if you use statistics, if you use mathematical tools, you can basically detect that there is a distinction between two separate pathways. But just looking at it, it's not obvious. So it's one reason why a lot of researchers in the field of brain networks come from mathematics and physics, because you need this analytical background to do some of the analysis presenting. So this is how the human brain looks like. So this is based on this non-invasive method, diffusion tensor imaging. So what you see here is the pattern of fiber tracts, and the color tells you the local direction of a fiber. So red means going from left to right, green means going from the back to the front or the opposite direction, and blue means going from the top to the bottom or the opposite direction. So you can see here the fibers going up and down the spinal cord here. You can see the corpus callosum connecting both hemispheres. And if we wait a bit longer, we can see the acute fasciculus, which is basically connecting language areas, Broca and Wernicke. And this fiber tract is relatively stronger in humans compared to non-human primates, which might explain better language ability in humans. So we can look at this network, and this can already give us some information about the subject. We can predict the age of the subject, plus minus 8 years. And we can predict the IQ of the subject, plus minus 3 IQ points. So just looking at this network can already give us some information on what is going on. And this is only the structure of the brain. So it's like looking at a map of motorways without looking at how many lanes are there, or well, looking at how many lanes, yes, but you're not measuring the traffic, right? You're only looking at the infrastructure, what roads are there in the brain. And this already tells you what capacity is there for intelligence and for different cognitive tasks.
Excuse me, can I ask?
Yes.
What do the different colours represent?
The different colors are the local direction of the fiber tract. Green means from the front to the back, blue from the top to the bottom, and red from left to right. Or the opposite directions. Yes.
On that note, can I just quickly ask?
Yes.
So I thought IQ was quite a subjective concept. Does that mean that IQ is like the definition of how many neural pathways there are in the brain?
It is related to that, yes. So basically, if you can easily travel through the network, you tend to have a higher IQ.
Right.
So you can basically predict that based on how easy you can travel through the network.
So you have studies of people who've taken an IQ test.
Yes.
And then you've measured the amount of neural pathways they have.
Yes.
And you've showed a direct correlation.
Yes. Yes. And you can also look at the network as a predictor of whether someone will get dementia later on. So basically, if you have higher flexibility, if you have lots of alternative paths to go through the network, you're more flexible to damage. So if parts of the network are damaged in the progress towards dementia, then other parts can take over. So this means that people with a higher IQ and with more years of education have a later onset of dementia. Because initially they can still compensate, but then the negative point is, well, once they get dementia, it goes downhill faster. Because by the time they see deficits, larger parts of the network are destroyed.
So you think the acknowledgement of knowing that they're having a deficit in memory loss accelerates the dementia?
It accelerates faster for this later onset, yes.
No, what it's saying there is that if by the time they discover that they have the dementia, so much damage has been done because it's been compensating for itself that whole time. Yeah.
You were talking about population averages here, aren't you? You're not talking about actual individual predictability of a single individual person.
Yes, you have an average error of plus minus 8 years of age, for example.
So hypothetically, if you measured my IQ or my connectome, you would not be able to say, you are going to get dementia by the time you're 65. But on the other hand, as a white man with a certain amount of IQ, you could say that from your overall population, you have a certain risk of getting dementia.
Yes. I mean, for dementia, it's never 100% prediction for you know, the progression. What we have here in the UK is the UK Biobank. It's unique worldwide, and it's the largest data set of brain connectivity. There are 500,000 subjects with genetic data and 100,000 subjects where imaging data is also being processed. At the moment, half of those subjects have already been measured, so 50,000. So we have data about 50,000 subjects. And this is really amazing. When I did my PhD, I was dreaming of one human subject. I couldn't imagine having 100,000 subjects in a database. So we have information about brain connectivity, but we also have information about diseases. So if someone gets dementia, for example, this will be updated in the online record, so you can go back and see, well, how did the brain look like 5 or 10 years before the onset of dementia? So that's very useful tool for the UK and internationally. What are some of the properties when we look at brain networks? One property is that there are quite a lot of long-distance connections. They are really crucial to integrate information from distant parts of the brain. We also have imbalanced connections. And this is something that is often forgotten in the field because the data we get about brain connectivity doesn't tell us the direction. So if we use diffusion tensor imaging and we know two areas are connected, we don't know whether it's a one-way path or whether it's bidirectional. However, if we look at animal studies where we can slice up the brain, we know that 15% of the connections are one-way streets. So presumably this is also the case for the human brain. We just don't know which ones are those 15% or maybe 10%. So those imbalanced connections can be very important. You could have a connection in one direction but not in the other, or you could have an imbalance where you have a very strong connection in one direction and then a very weak one in the other direction. And this gives us more degrees of freedom, so we have more parameters to tune. If we only have connections going both ways with exactly the same strength, then, well, there's only the strength, total strength to play with. And if we have imbalanced connectivity, we can make one connection stronger, another connection weaker, and so on. And this means we can have more kinds of behavior in the network. Another feature of networks, and that's not just for the brain, it's for all networks, is that you have a modular organization. So modular means that you have many connections within a module, let's say within the web module here, but few connections to other parts of the brain. And this helps to focus on one particular task. So you might have this part of the brain dealing with visual information, and this part with acoustic information, with sound. And you don't want them to interfere. Okay, so you want this part concentrating on vision, and having a modular organization helps because you don't get all this noise from other parts of the brain. You can just focus on processing vision. Finally, the other architecture we see is that some nodes are highly connected. They're called hubs. So this means that you might have, on average, 10 connections for each brain region, but there might be one brain area with 100 connections. So it's basically a lot better connected than other parts of the brain. And this means that either this region can integrate a lot of information from other parts of the brain, or it can broadcast, it can spread information to a lot of other places. And we also have a witch club network in that there are stronger connections between those hubs. So this means that you might have one module over here, the hub is integrating all the information from that module and then distributing it to another hub that can then distribute it to another module in the brain. We also know that a lot of brain diseases are linked to changes in hubs. So for dementia, for example, you have hippocampus that shows changes, and for other diseases, you have other hubs in the system that are affected. So if something goes wrong with the hubs, it usually has a very bad effect, because a lot of information, a lot of traffic is going through those hubs. So it's like locking out London Heathrow. It would affect the flights in the UK, for example. Unless they're on strike anywhere. So just to give you one example of changes we can see for brain diseases. So each brain disease shows some distinct changes, some distinct nodes or edges that are affected. Just one example here would be Lewy body dementia. It's a rare form of dementia. It's like 5 to 10% of the cases, whereas most people have Alzheimer's disease. For Lewy body dementia, some patients have hallucinations, visual hallucinations. That occurs in 80% of the subjects. It could be featuring people, animals, or objects. And it's highly diagnostic, so it's very specific to Lewy body dementia in contrast to Alzheimer's disease, for example. There are some ideas what might happen, so it might be associated with cholinergic dysfunction. But then the question is, where exactly in the network does hallucination come from? And why do some Lewy body dementia patients show hallucinations and not others? So we looked at the connectivity of the brain, and we looked at connectivity between different parts of the brain. We found that there's one particular connection that shows changes for this patient group. So you have the number of fibers between the cortex and nucleus basalis minor, so one subcortical brain structure. And for the patients that have visual hallucinations, you have a lower number of fibers compared to the ones that do not show hallucinations. So we can basically pinpoint the change in the network, and we can say, well, this particular fiber tract is affected and is then leading to hallucinations in those patients. We can do that for many other diseases as well. So we can get some idea which parts of the brain are affected. So what are interventions for connectome disorders? So one possibility would be to change network activity so that episodes of bad behavior do not occur. So for Parkinson's disease, patients have tremor, so shaking of the hand, and you can basically implant a device into the brain that is electrically stimulating one part of the brain and then reducing the tremor and ideally avoiding a tremor in patients. Another example is non-invasive stimulation for Tourette syndrome. There's currently a trial here in Nottingham led by Steve Jackson from School of Psychology, and he's also leading a startup company, New Therapeutics. They have a small device that is on the wrist and that is stimulating the wrist and is then reducing the amount of tics in Tourette syndrome patients. So that's one possibility to basically change activity in the system. But what I want to do in the future is basically change connectivity in the system. So change the architecture of the network to make sure that the bad behavior of the network is not occurring in the first place. So one example would be avoiding psychotic episodes for schizophrenia patients. So changing the network in a way that brings it closer to control subjects and avoids the occurrence of those episodes. So if we look at how the brain is developing, we have the early age, and then you have development of healthy brains, but then you have some changes during development that lead to different architectures, leading to depression, schizophrenia, and so on. And the idea is to have computational models to predict what is the best strategy to get the system back to normal or closer to normal in order to avoid the symptoms we see in patients. A treatment could be non-invasive stimulation, it could be surgery depending on the disease, or it could be what pharmaceutical drug to give. But then one question is, if we want to change connectivity in the brain, what fibers and what nodes of the network should be targeted? So what is stopping us from having more fine-tuned brain stimulation? As I mentioned, one question is what part of the brain to target. Depending on how you divide up the brain, you might have 100 regions, you might have 300 regions. It depends a bit, but let's say you have 100 and you have more than 1,000 fiber tracts in the brain. So how do you decide which one to target? So that's one challenge. Once you know where you want to target, how do you target? So what is the optimal stimulation protocol? For how long should you stimulate, with what frequency, and what amplitude, strength, and so on? Another challenge is to estimate long-term effects and unwanted negative side effects. Concerning long-term effects, if you currently have an implant, so in this case, we have an implant for deep brain stimulation and major depression, what happens is that initially the implant might work, but after a couple of weeks it doesn't work, because the brain is changing as a response to the stimulation. And then you have to readjust your stimulation parameters. So it would be better to have a computer model that tells you what are the correct parameters, what are parameters that are working, rather than using trial and error to readjust the system. And also you want to avoid a negative side effect. So you don't want to have brain stimulation that leads to negative effects weeks or months down the line. And then finally, you would really like to have non-invasive approaches. Even for Parkinson's disease, there were very few patients who have a brain implant to stop tremor. Even if you think about cochlear implants, which is relatively non-invasive compared to big brain surgery, less than 6% of eligible subjects agree to have a cochlear implant. So people really don't like implants. So it's really important to develop interventions that are non-invasive. In order to do this, we developed a way to look at the brain in higher resolution in order to find out where exactly to stimulate, so which parts of the brain should be targeted. So what you see here is zooming in and out of the whole brain. I just try to let's do it like this. So you basically have the left half of the brain in the upper left corner, the right half of the brain in the lower left corner, and then we can zoom in. So we can zoom in, we see individual brain regions labeled on the left. But then we also see that we see a network architecture within individual brain regions. So we see different modules in those regions here. And this gives us better information where to stimulate. We're not just sticking the electrode somewhere into a brain region, but we could say we want to target one particular module within a brain region. So here you see the pattern of modules within brain region for two regions, a left superior parietal and left superior frontal. You can see the connectivity within those regions and then connectivity between the regions. And those boxes in the middle are different modules within a brain region. So if we look at the connectivity between those two brain regions, it's not just random, but we see that a module in one brain region is connected to a module in another brain region. Okay, so those fibers tend to be in those crossings here. And there's also a spatial pattern for this. So all those modules within brain regions are spatially nearby. So you can see that if a part of the surface belongs to the same module, then it's just the same patch of tissue on the surface. So we're currently testing whether, yes?
Sorry, I just want to display my lack of connectome by not understanding what you said.
Oh, okay. So basically, you have two brain regions, okay? And within each brain region, you have modules based on the connectivity. And what we find in the middle there is a module in one brain region is connected to a module in the other brain region. And this is showing you where those modules within a brain region here, where they are on the surface of the brain. And so the colors give you the different modules, and you can see that those modules are just patches of tissue on the bone. We're currently testing whether there's a master student working on that at the moment, whether those modules we see in the network also have functional significance. So if you think about parts of the brain for movement, within that brain region for movement, there might be one module for arm movement and another one for leg movement. Okay. So we're currently testing that, whether that is the case, whether those structural modules correspond to function as well. And we then wondered whether this high resolution or super resolution connectome can also give us information about brain diseases. And for that we looked at epilepsy, in particular temporal lobe epilepsy. For temporal lobe epilepsy, very often the drugs don't work and then patients have to undergo surgery. So we have very detailed information about those patients. If the drugs don't work, then patients wouldn't show up in an MRI scanner. But the drugs are not working, so you have MRI scans to plan surgery, and we have that type of information here. So we take information about the surface, information about connections between different parts of the brain, and we reconstruct the low resolution network. So this is just connectivity between different brain regions. And the high resolution network gets connectivity within brain regions. Just skip this one here. We can then look at the pattern of connectivity within one individual brain region, and we can check whether this connectivity is different from that of healthy subjects, if we look at those epilepsy patients. So we can check, is it different and how different is it? And we find that those local changes within brain regions increase over time. If we have a lower duration, which means patients had epileptic seizures for fewer than 20 years, we get a pattern where relatively, well, not really small, but smaller compared to longer disease duration. So you have several regions that show those local changes. But if you look at patients that had epileptic seizures for longer, for more than 20 years, you see this pattern on the right-hand side, where first of all you have more brain regions that show local changes, and also those local changes become stronger, as you see indicated by color and the size of the sphere. So one question for us now is, do those local changes appear before the global changes? So for example, for schizophrenia, can you predict that someone will have a psychotic episode 5 years before based on local connectivity? This is something we're currently testing with data from Shanghai to see whether people who are at risk of developing schizophrenia, where we know whether they're developing schizophrenia or not, whether we can predict that in a computer model based on this local information. So we're currently testing that for schizophrenia, but it might be of interest for other diseases as well. We can also look at those local changes and compare them with the outcome of surgery. The sad part about epilepsy surgery is that it doesn't work in 1/3 of the cases. So you remove parts of the brain, relatively large parts of the brain, and in 1/3 of the cases there is no improvement for the patient. So how does the network differ? So this is just looking at the network before surgery. It's all based on the scan before the procedure. If we have a good outcome, then you see that fewer brain regions show local changes before surgery. If there's a bad outcome, there are more regions that are already affected with those internal changes. So the more epilepsy has changed the brain, the less likely is it that surgery will work. We can then use machine learning, so we can then use, yes?
Sorry, I have a question. Could that be a consequence of the use of astrocytes? Because when you remove a part of the brain will grow astrocytes that tend to reestablish the original connections. And because that part of the brain has been active, working in a specific way for so long, that most likely when you will get the astrocytes coming into the brain, they will most likely attempt to reproduce with less readings. Could that be also?
So you will not reproduce fiber tracts, but you do have a way that there are other parts of the brain that are not involved in seizures before, or you don't easily see them. But then after one part of the brain is gone, they become more active. They get disinhibited and then they cause new seizures. So that's the basic idea, that because there are more parts of the brain affected, then it's easier for epileptic seizures to restart. So we can use computer models to predict whether surgery works or not. This will be very useful for patients, in particular the third where it doesn't work. What you see on the left-hand side is the performance if we look at local connectivity, the temperature warning as well. So anyway, we have the network of local connectivity, so connections within brain regions, and then we have the standard approach to use connectivity between brain regions. And we see a better performance for within region connectivity. So basically, the more of this box is filled, the better the performance. But in particular, the specificity is increasing. And this tells us the likelihood of predicting bad outcomes. So if we take the standard approach, in 75% of the cases, we can correctly predict that surgery is not working in a patient. But if we use local connectivity within a region, we can predict that in 90% of the cases. So that's promising. So it might be useful in the future before brain surgery to have a quick look at those measures. So are local network features better predictors for what regions should be targeted in surgery, or possibly also for invasive and non-invasive brain stimulation in other diseases? So looking at those local changes might also tell us which part of the brain to stimulate in a patient. There will be even better information in the future. So Nottingham was awarded some funding to establish a high-field scanner, and this gives us higher spatial resolution. So normally you would have 1 millimeter resolution if you do an MRI scan, but you can basically get much better resolution below 1 millimeter with this new type of scanner, 11.7 Tesla. So that's coming to Nottingham, and it will be a national center for imaging here in the UK, the first time to have this high-field scanner available in the UK. So we will get more information about local connectivity. We will be better able to understand what local changes are going on. So how do we change connectomes? So this is the need for personalized, targeted, and non-invasive approaches. There is a new method to stimulate the brain, and this is focused ultrasound. You all know ultrasound from pregnancy scans in the hospital, for example. So you can use the same ultrasound in order to stimulate the brain. You can either increase or decrease activity in the brain, depending on your parameters. If you turn it to 11, you can destroy tissue as well, but that's not the equipment we have here in Nottingham. But if you spend £1 million and you get 1,024 sound sources rather than one, you can actually destroy brain tissue, and this is used in clinical trials to destroy brain tumors and also to help Parkinson's disease patients. But we are not doing that here. With the device we have in Nottingham, we can basically stimulate directly deep brain structures. So we are one of 4 current centers in the UK having this device. There are other centers, of course, in the EU internationally. And the benefit of using focused ultrasound is that you can directly stimulate deep in the brain without stimulating anywhere in between. So the sound source is going through the brain, but it's only at the focus that you have enough energy to increase or decrease activity in the brain. It can also be very focal, so depending on the device you have, you could even go down to a few millimeters. You can target deep brain structures, which is really interesting because a lot of brain diseases really involve deep brain structures. So it's interesting to be able to stimulate them. You can increase or decrease. The device can be used for humans or can be used for rodents, animal studies. And it's also possible to measure with EEG or MRI at the same time. So you can put someone into an MRI scanner, you can stimulate with ultrasound, and you can measure directly how activity in the brain is changing. So what does ultrasound do? It looks a bit like that. You have the sound source outside the skull, and you can see it's basically focusing here. And then you have one target structure that is increasing or decreasing in terms of activity. This lasts for 2 hours. Sometimes it can last for, basically in rodents, in mice, people have studied it for longer. You don't want to put humans into an MRI scanner for a long time, that's the limit. But for rodents, we know it lasts for 8 hours. So you can see effects and changes in brain activity. And what actually happens is that there were some parts in the membrane of the cell, those here, and you have a mechanical deformation because of the sound that is traveling through the tissue. And you have some channels that are mechanosensitive, so they basically react to mechanical pressure. And if there's enough pressure, those channels are opening. This is then leading to changes in internal calcium, and this is then leading to opening of other channels and leading to changes in brain activity. Most of the studies appeared in the last 3 years. This is a very novel area. In 2019, the first international conference on brain stimulation of ultrasound happened. There were around 100 participants in Oxford. And now more and more people are getting interested and starting to use ultrasound. What I show here is just an overview of studies in human subjects. You can see that different parts of the brain have been targeted. There have been studies in healthy human subjects and in different patient populations. We have temporal lobe epilepsy as one example, traumatic brain injury. And there were studies in depression patients that have mild or moderate depression, and you get an improvement in mood for those patients. And you also get an improvement for mood for healthy subjects. So if you want to feel better, just buy an 80,000 pound focused ultrasound device, and you feel better for at least 2 hours. So what we're doing in our lab is this current study here, which is looking at effects in rhesus monkeys and in healthy human subjects. But the hope is to move on to patients sometime next year, depending on funding and so on. But we're currently testing this in healthy human subjects. We have data from collaborators in Newcastle and in Lyon. They stimulated in rhesus monkeys, so we can look at how the brain of those monkeys is connected, and we can look at the effects in terms of brain activity over time. And we want to do a study in humans here in Nottingham to look at changes in brain activity in humans. And part of the project is to develop a computational model predicting what the effect will be of ultrasound stimulation. So we have the connectivity between regions, we have a skull model, and we have parameters for stimulation. And based on this, we have a computer model to see what kind of changes would we expect over time. And we then want to compare that with the actual changes we observe in monkeys or in humans in terms of fMRI activity, in terms of correlations, and in terms of time course of changes. So this is some first result for rhesus monkeys, where you have stimulation in the left hippocampus, so in one deep brain structure. So that's the stimulated part of the brain. and you can see increases and decreases in terms of how you have linkage to other parts of the brain. So this linkage is not a change in structural connections. It's not like the fiber tracks are changing. It's looking at whether you have correlated activity. So if you have two brain regions that show the same activity pattern over time, you would say there was a functional link between them. And increase means the functional link gets stronger, so they are more similar in terms of their activity, and decrease means that they become less similar in terms of their activity. So we see that you have widespread changes in the network, and we're currently looking into the time course in more detail. If brain regions are closer to the left hippocampus, to the stimulation site, those changes are stronger, and you also have a higher duration of effects. So we're currently developing computer models to basically predict what the effect would be in both rhesus monkeys and later on in humans. So in summary, brain and mental health conditions show distinct network changes. So we can basically look at the brain of a subject and identify particular connections and regions that are affected by a certain brain disease. And we are just starting to use this to inform interventions. I was telling you about brain surgery, so looking at connections and informing whether surgery works or not. But you might also use it for deciding which drug to give. So for epilepsy, for example, it takes some time before you find the right drug for a patient. And looking at the network, you might get an idea what drug should be used for that particular patient. It might be useful for other diseases as well. And connectome-based computer models can use to predict targets for interventions. We can take brain connectivity and use computer models to predict what would be the effect of an intervention and what is the best intervention for one particular patient. And there was a need for non-invasive, targeted, and personalized interventions to deal with brain network disorders. And focused ultrasound fulfills this requirement. It's non-invasive, you can directly target structures and change activity, but in the long term also change connectivity so that those dynamics don't occur anymore that are causing symptoms. If you want to learn more about how the brain is connected and about how the brain changes for different brain diseases, this is the latest book in the field, 2020. So it talks about the evolution of the brain, so how do we get from simple organisms like worms, how do we get to the human brain? What is changing during evolution? What is changing during brain development? And what is changing for different brain disorders? What changes after learning? And what changes after brain stimulation? There are lots of things in there. And finally, I would like to thank my team here at Nottingham and the former team members, collaborators, and funders. If you want to follow this kind of research, you can follow me on Twitter, Connectome here. So thanks for your attention. Maybe that's the point about it. Yeah.
Thanks for your response. Any questions?
Hi. You showed some Connectome data of audio brain when the connection are either really important. Have you or your group or researchers started to measure how the connection are following through while you grown up, like from kids to adult age stories? I know that it's more challenging from MRI quantitative motion analysis.
There was such data, so the problem is always to get a study funded that runs over 10, 20 years. But there were some longitudinal data sets where you have the same subject measured over a couple of years to look at how the brain is changing. It even gets better. There was a project at UCL and King's College. They look at connectivity before birth.
Okay.
So it's called Developing Connectome Project. So you put like a pregnant woman into an MRI scanner, and then you can basically measure connectivity before birth, so you really see how connections are growing. So there are more data sets that are available at the moment.
Alright, so like, I don't know if it's a thing to say, but plasticity is the process to form this connection. When you got neurodegenerative disease, you can say that like it's a kind of progress process, or we don't know yet.
Yeah, it might not be exactly. So in general, of course, connections go down. So even for healthy aging, if you don't have Alzheimer's disease, you still have a loss of connections over time, and then leading to cognitive decline over time. I'm not sure whether it's exactly the same pattern as during brain development. Something to check.
More biologically probably than
Yes.
in imaging. Alright, thanks.
Thank you for the talk. Has any research been done into, say, like patients with autism, for example? So can you distinguish brain connectivity between people with autism and typical brains, for example?
Yeah. So there were some differences for autism. I'm not aware of the details of the differences, but there were studies looking at autism. One funny thing is ADHD. So a colleague of mine looked at ADHD and looked at two data sets, one from China and one from the US. And for patients in China, there was a clear difference in brain connectivity between patients and controls. And for the US, there was no difference. Well, there was some difference, but not as strong as in China. And the reason is that you get healthy children who are diagnosed with ADHD in the US in order to get Ritalin and perform better at school. So that's the reason why you don't see differences between patients and controls. So it's so for ADHD, it's a bit mixed depending on what country you look at. But with autism, you do have clear differences.
How do you define normal brain?
Well, you don't really define it. You always do the comparison of control subjects and patients.
How do you know what? Down the line.
Yeah. But it's always a challenge. So of course there are lots of studies where healthy control, or in general, the study is like psychology undergraduate students, because as part of their studies, they have to take part in research studies. And of course, they are not representative of the population. So it's one challenge in the field to get better controls. Yeah.
Thank you. The focused ultrasound stimulation or modulation of pathway activity, how long do you have to apply that on the phone to get your 2 hours of activity? So that's one question.
Yeah.
It's great if I have Parkinson's and I can ensure myself 2 hours to go to the party and not do it in good way. But I suspect that when we use this clinically, we will then have to have recurrent sessions. Has anybody looked to the body adapting to the stimulation? Because it's our chemical pathways that's actually motivated, they're acting all that. So the body tends to adapt and become resistant. Has anybody studied that recurrent stimulation for those ultrasound?
Yeah.
Does it result in a change in frequency or increase in whatever?
Yes.
Thank you.
So for ultrasound, no one has done it yet, because it's really, really novel. People are not really sure how long the effect lasts either, because you can't put humans into a scanner for 8 hours. It's a bit uncomfortable. So effects might last for 8 hours as in rodents, or they might even last for 20 hours. We don't know. And coming back to your question, how long? I wonder if this slide is still. Sure, the slide is. No, sorry. So basically, stimulation is very, very short. So you only stimulate for a minute, less than a minute, and then you get those long-lasting effects. It's very, very short stimulation. Okay.
Hi, thank you for the talk. So you mentioned the DTI and the dye injection as a measure as a way of actually viewing the connectivity in the brain. However, it's more of a yes or no answer whether we could connections there or not. However, what about the dynamical features, like, I don't know, differences in signal propagation or synaptic strength or count of fibers or, I don't know, neuronal populations? Is there a way of actually looking into that rather than just say, yes, the connection's there?
Yes. So there are some ways to get more information, not necessarily about activity traveling over the fiber as you stick an electrode into it. But you can get information about the number of fibers that goes between brain regions. You get some information about the isolation of myelin. And this gives you information how long it takes for the signal to propagate. So if you don't have this myelin, this fat isolation at all, you only have 1/10 of the speed. So you only have 1 meter per second in terms of the signal. Whereas if it's myelinated, you have 10 meters per second. So you do get some more information that you can use for diagnosis and also for computer models.
Just to talk about, what about, you know, people who suffer dynamics of demyelination? So do their brain show different structures?
Yes, and this is one thing we want to test with ultrasound, whether we can basically increase the strength of the surviving fibers. So there are a lot of diseases where cells are dying and then the axon is dying as well. So maybe only 50% of the axons are still there. And what we want to try with ultrasound is, can we actually increase the strength of those connections? So if we can double the strength of those connections, that can maybe compensate for the half of the connections that is missing.
So you're not just destroying them?
Yes. Yes, at the moment in a computer model, so we haven't tested it yet in subjects.
This is a really naive question, but is it always the more connections the better? So then in these cases, they always just get reduction?
Most of the time, yes, because reductions are linked to cell death. And that's part of many diseases. I guess the only exception would be epilepsy. So basically, the stronger the connections are, the worse, because if you have strong connections, it's easier for a seizure to spread from one point to another. I think that's maybe the only exception, but normally we see reductions.
More naive question to some of it. So, maybe you can tell me a little bit about plasticity. I'd like to know if neurons die, they get replaced, they grow more of them. What is the story around death and, you know, birth of neurons? And the other question is, you showed us a three-dimensional map of the brain, and you said that if you see the connections and the number of connections, you can tell if somebody is smarter or not so smart. So, can I take this test and, you know?
Coming to the last point, so the problem is that an MRI scan costs 500 pounds. A paper IQ test is like £1. You know, so, but it's possible.
But I'd like the picture as well. And what about the plasticity of cells? We are computer scientists, and we use fixed size neural networks. And we are thinking that maybe this isn't right. Maybe we need to start small and grow the network as we go.
Yes. So basically in the brain, you have some parts of the brain where you see that new cells can be generated. But for most parts it doesn't happen. So if a neuron is dead, it's dead. But it's like in computer science, graceful degradation. So you might knock out large parts of the system before you see an effect in dynamics. But there was one peculiar thing about the brain, which is, to my knowledge, not really realized from machine learning, and that is functional compensation. So you basically have different parts of the brain, and you would say, well, this part of the brain has this particular function. But in reality, 10% of the neurons also do something else. And so what happens and that's an animal study in a cat, you basically destroy one part of the brain and you find that one other part of the brain is taking over the function. So basically, you know, all the visual functions are almost fully restored. And what happens is that before destroying this part, 10% of the neurons have a similar function to that area. And then afterwards, 50% of the neurons have a similar function. It's upregulated. And this makes the network very robust. So in terms of computer networks, you could have some machine learning where you not just have one part of one function, but you have some overlap in function, which makes the whole system more robust and also easier to adapt to different kinds of input. So it would be one idea to test. Yeah. And it was one idea tested by colleagues Herbert Jäger, echo-slave networks. So basically, we know that there's a modular organization in the brain, and if you start machine learning with a modular network, you get better performance than if you start with a random network. So that's another way where you can get information how the brain is organized, and it leads to improvements for artificial neural networks. Okay.
Most brain diseases like schizophrenia and Alzheimer's and such like are clumped together diseases, and in some cases in schizophrenia, very different types of schizophrenia. Is this type of research actually teasing apart some of these diseases that look like schizophrenia or whatever, and aren't actually, because they don't belong to that same?
Yes. So that's one reason why I talk about computer models. So we're not just looking at the network, but we also simulate what behavior the network is showing in the computer. So you might have two networks that look very different, but they show the same behavior. On the other hand, you might have two networks that look quite similar, but the behavior is very different. And we do that in order to have personalized models to really understand what is going on in one particular schizophrenia subject. Because the pattern will be very unique for that subject. There's one reason why your problems with drugs, they don't work consistently, or stimulation doesn't work consistently. So we really need a personalized model for each individual patient. That's what we want to do with our computer models.
But is that potentially going to show us that we need new names for new diseases that we're going to find? We've labelled schizophrenia, but in actual fact aren't schizophrenia. They're a new disease.
Possibly, yes. I mean, the disease classification at the moment is not very useful. So
that's what I need to
Yes. So there might be another classification. And in my opinion, it wouldn't just be based on the network, it would be based on the behavior of the network. And that can be used to classify. Yeah. This will be something for the future to really use networks for disease classification.
You mentioned the region brain responsible for regeneration of neurons. I'm pretty new to neuroscience, so what region is that called?
Region related to
neuroplasticity, regeneration.
Yes.
Of neural pathways.
Yes. So the example I was given was just one particular example where area 17, 18, and 19 of the visual cortex are knocked out, and then TMNS is taking over the function. But it's a generic principle that other parts of the brain have some functional overlap.
It's quite inclusive.
Yes, it's a generic one, yes. Yeah. It's not one particular region.
And also, earlier you mentioned there's the differences between a neurotypical versus neurodivergent brain. Do you maybe expand on what the differences are?
Yeah. So difference we see are in reduced connectivity. There are differences in terms of how easy information gets spread, and also how long it takes information to spread, because the delays in the system are really, really important. So for the visual system, for example, you have, so let's say you have a wet cup on the table here, and there was one part of the brain that is recognizing that it's a cup. Another part of the brain recognizing the color red. Okay. And another part of the brain recognizing the position of the object on the table. And we know that it is a wet cup on the table because those different parts of the brain encoding for wet and for cup and for on the table, they're active at the same time. Okay. So you have synchronous activity, and this is indicating for us that it's a wet cup on the table. But the problem is that if you have changes in brain connectivity, it could be demyelination, so you get rid of the isolation around fiber tracts, and then it takes a lot longer for information to spread. It could be the loss of fiber tracts or fibers, and this means that information doesn't arrive at the same time. And then you basically have cognitive problems if you don't have information arriving at the same time. So those are some changes you could see in terms of brain connectivity.
Cognitive problems, you say?
Yeah.
And just ask a
Yes.
question. How do you get healthy volunteers to volunteer for?
So at the moment it's members of our lab who are just testing the device. But there have been a lot of safety studies in the field for ultrasound. So there have been studies where they turn the volume to 11. So we're not doing that in our studies, but they turn the volume to 11 and they check for brain damage and they don't find brain damage in the tissue. So it is a safe procedure.
Any other questions? Just over here, that far one there.
The connectomes that we've been looking at, for example, the one on the front of this book, that's computed from kind of MRI data, is that right?
Yes. Yeah.
So how reliable is it to use MRI data to construct these connectomes? Has there been any studies where they'll compare this connectome against, for example, the dye method that you mentioned?
So one part of this is that there were retest methods where you measure the same subject a few days in between. For this structural connectivity for the fibers, it's very, very consistent. It's different for fMRI. If you look at brain activity, you get different brain activity in the morning and the afternoon. So just something about waking up, getting from the horizontal to the vertical, the water content in your skull is a bit different, and you get different patterns of activity. But if you look at the connectivity pattern here, that's very consistent. There were studies. Actually, we had read that one. Well, 1 or 2 years ago, we had a study in rhesus monkeys where we compared data based on injected dyes and based on non-invasive diffusion sensor imaging. There was a high similarity between them.
And that's one final question. There are two actually. Is your book available on Amazon?
Yes.
And
This link goes directly to Amazon.
Amazon, excellent. And can we get this video? There's lots of stuff in there, and I'd like you to listen to myself.
Yeah. So there's a green light, so I'm very hopeful that everything was recorded. And if yes. Oh, okay. Well, if it was recorded, then it should be on the public lectures website later on.
Thank you.